Hi everyone! For our class field trip, we visited two places. Yes, Taman Botani and Taman Wetland Putrajaya. And here a quick look at what we did at the wetland. Taman Wetland is a Malaysian first man-made freshwater wetland. It's not just beautiful, it helps filter water, reduce flood, and provide a safe space for birds, fish, and other wildlife. When we arrived, we gathered in the hall for a short briefing while enjoying breakfast. We learned how the wetland system works and why it's so important to the environment. Then, we went outside to start our activity. Using scissors and tape, we explored the tree to identify different wetland plant species. It was so fun and hands-on, like a scavenger's hunt. And along the way, we discovered how this plant help clean the water and support the ecosystem. It was short but meaningful experience. Definitely something different for learning in the classroom.
Assalamualaikum and hi. Now I will presenting practical 1, basic concept of statistic and variation. The objective of this practical are to calculate mean, variance and standard deviation, and to understand the correlation between leaf length and width. We measured 30 leaves from 3 different species, divided into 3 sets. Now I will present the results starting with the set 1. For set 1A, which is the leaf length, we measured 10 leaves from Garcinia ultraviridis. The mean length was 17.4 cm, which is the highest among all 3 sets. The variance was 1.77 and the standard deviation was 1.33 cm. This showed that the leaf length in set 1A were quite consistent and not very spread out from the mean. Okay, next for the set 1B, which is the leaf width from the same species. The mean width was 6.1 cm, the highest among all the sets. The variance was 0.27 and the standard deviation was 0.52 cm. This means the leaf width in set 1B were very uniform and close to the average. Next, for set 2A, we look at the leaf length from Neobalanocarpus helmi. The mean length was 15.31 cm, which is lower than set A. However, the variance was 4.61 and the standard deviation was 2.14 cm, the highest among all the sets. This tells us that the leaf length in set 2A were more varied and not consistent. Okay, for set 2B, the leaf width had a mean of 4.71 cm, which is the smallest among all 3 sets. The variance was 0.76 and the standard deviation was 0.87 cm. So, although the leaf were smaller in width, there was still a fair amount of variation. So, let's move on to set 3A. We measured the leaf length of Hopia odorata. The length was 11.96 cm, the shortest compared to the other sets. The variance was 1.2 and the standard deviation was 1.1 cm, showing that moderate variation but with consistent size range. So lastly is in set 3B, which is the leaf width. The mean was 4.87 cm and the variance was 1.19 and the standard deviation was 1.09 cm. This showed that although the leaves were small, the width had moderate spread from the mean. So let's move on to the discussion. From this practical, we learned how to apply basic statistics like means, variance, and standard deviation to analyze biological data. We collected 3 leaves in total, divided into 3 different sets from different 3 species. Set 1 had the largest leaf with the highest mean length and width. Set 2 had medium-sized leaf, but it showed the highest variance and standard deviation in length, which means the leaf size were more varied. Set 3 had the smallest leaf, with more consistent size compared to set 2. By comparing all three sets, we can see that leaf size depends on the species and the natural variation exists even among leaves from the same tree. This practical helps us understand how statistical tools can be used to describe and compare biological characteristics. So let's move on to post-let question. For the first question, we compare the mean value of leaf length and width between the three sets. Set 1 had the longest and widest leaf with a mean length of 17.4 cm and width of 6.1 cm. Set 2 had a shorter mean length of 15.31 cm and width of 4.71 cm, while Set 3 had the smallest leaf with a mean length of 11.96 cm and width of 4.87 cm. This shows that different species have different leaf size on average. So the answer for question 2 is variance is a value that tells us how much the data is spread out from the mean. If the variance is high, the data value are more spread out. In this practical, we can see that set 2 has the highest variance for leaf length, which means that the leaf size were very different from one another. Standard deviation is the square root of variance. It shows how much the data differs from the average. Using the same unit as the measurement, which is centimeters in this case. So for the last question is, we use different sets of measurement to make the result more accurate and reliable. Using more data from different sets helps us to reduce error and it gives us a better understanding of natural variation in leaf size.
Hi everyone, me Nurul Imana Sohaben Zakaria. For Practical 2, we carry out a quadrat sampling activity at two different sites in the Botanical Garden Putrajaya. First, the objective to determine the plant species diversity and density at both sites using the quadrat method. Next, the method. We use a 5 meter by 5 meter quadrat at site 1 and site 2. All plant species inside each quadrat were identified and counted. Then, we calculated the density using the formula total number of individuals divided by the area of the quadrat. So this is our result. Site 1 had 6 species. The most common were Dipterocarpus oblongifolius and Dryobalanopes aromatica. Site 2 also had 6 species but with different dominant one, which is Areca catechu and Antidasma rasambila. Only one species, Dryobalanopes oblongiphobia, was found in the blue site. To sum up, the two sites had similar species number but different type of plant. This may be because of a difference in the sunlight, soil, water or human activity. Quadrat sampling helps us see how plant distribution can change even in nearby area. So moving on to the post-let question, what is a quadrat? A quadrat is a square tool like a 5 meter by 5 meter used to count plant or non-moving organism in one area. It helps estimate species and biodiversity. Next, the two basic rules is place the quadrat randomly or in a pattern to avoid bias. Also, use the same size and shape for all quadrats to keep data consistent. The pros and cons of the quadrat size is, for a large quadrat, more species, better data but slower. For the small quadrat, faster and easier but might miss some species. For question 4, the main problem is the large habitat like forests or water area are hard to access. Plants may be hidden, unevenly spread or hard to reach, which make accurate data collection difficult. And for the question 5, because each area has different conditions like sunlight, soil, water and human impact.